ReactLab KINETICS is designed to analyze multi-wavelength chemical process data. It is ideal for analyzing data collected using photodiode arrays attached to stop flows or other kinetic instruments. The aim is to analyze the underlying kinetic mechanism and determine the rates and equilibrium constants for each reaction step. All data, reaction models and results are held in pre-formatted Excel workbooks. ReactLab interacts with these workbooks to perform the analysis. To demonstrate the procedure for analysis, I will start with an empty kinetics Excel template. This is the main worksheet where the model will be entered. It is currently empty, as is the datasheet. I have some kinetic measurements which I will cut and paste into this worksheet. Here are the data, a series of spectra measured as a function of time. The individual spectra are in rows with time down the vertical axis. I will now copy this data with the time and wavelength information to the template workbook. You will notice that Excel has automatically populated these grey reference cells with the total number of times and wavelengths in the dataset. The next thing to do is to save the workbook. To conduct an analysis, it is always necessary to load the populated workbook from ReactLab once it is running. This is the main ReactLab GUI. The first thing to do is to load the Excel file we've created. And here is the data we pasted in. ReactLab automatically creates an interactive 3D display of the data. I'll put this to one side for the time being. If I now return to the main worksheet, I can enter a reaction model that I think is appropriate to fit to the data. In this case, I'm going to fit a simple consecutive A to B to C reaction. So I type the model symbolically in the model editor. I can also provide an appropriate label for each parameter. In this case, K1 and K2. Now we are ready to compile the model. To do this, I press the Compile Model button in the main GUI. ReactLab reads in and interprets the mechanism, identifying all the steps and species names, which it then outputs to key fields in the workbook. At the same time, ReactLab translates the mechanism into an internal coefficient representation for use in the modelling and fitting computations. Before fitting, we must provide estimates of the nonlinear parameters, in this case the two rates K1 and K2. I will put in 2 per second and 0.2 per second. We also need to provide initial concentrations. I'll put in 0.01 molar for A, 0 for B and 0 for C. We also specify whether the spectra are coloured, non-absorbing or known. If we have known spectra, we need to provide those spectra in the auxiliary sheet here. I will specify coloured spectra which will be fitted during the analysis. Finally, I need to specify which of the nonlinear parameters I wish to fit. In this case, I'm going to select both of the forward rates. I now return to the ReactLab window. Before actually conducting a fit, we can use the update function to see how well the model, with these initial parameters, matches the data. We can see that the match is not perfect. There are some systematic deviations in the residual surface, and I am only expecting single peak spectra for each species. Nevertheless, this is clearly a reasonable starting point for the fit itself. The fitting is iterative, and as it proceeds we see the square sum reduce and converge to a minimum. We now have a completely random residual surface. The species spectra are now all single-peaked as anticipated, as a consequence of the now optimized concentration profiles. We can now use the ReactLab Graphics GUI to take a closer look at these results. We can superimpose the modelled Y-count values on the data. We can select single traces for inspection on their own, or we can scan through the series of spectral measurements continuously. I can also easily switch to the time dimension to see the kinetic profiles at each wavelength. Other options include the ability to view the time base on a log scale, or we can adjust axis labeling, print out the graphics or copy the graphics to the Windows clipboard for insertion in other documents. All the optimized parameters and their errors, together with the square sum, have been written back to the workbook. The best fit concentration profiles and spectra have also been written back to the results worksheet, where it is now possible to use Excel charting tools to display the results. Of course, any post-processing of data or results, or charting in Excel, can be done quite independently of ReactLab. Having completed the analysis of this data, we can now close the Excel file. The user is always prompted to save the workbook if its contents have changed. It can be reloaded at a later date and will resynchronize with ReactLab, allowing a seamless continuation of experimental analysis with all previous settings preserved. That completes this introductory tutorial. Thank you very much for listening.